This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 211, recorded on December 13th, 2012. Hi, everyone. I'm Vincent Racaniello, and you are listening to TWiV, the podcast all about viruses. This episode of TWiV is sponsored by ASM, the American Society for Microbiology, the world's largest membership society for microbiologists. Find out how ASM membership can help advance your science, your career, and your network. Go to asm.org/advance. Joining me today from Ann Arbor, Michigan, Kathy Spindler.
Hi, Vincent. How are you?
I'm fine. It's an unusual hour we're recording here.
Mm-hmm.
You're an hour behind us, is that right?
Oh, we're same time, 10 o'clock.
10 a.m., and it's a different day, Thursday. I just feel weird. I'll get over it, though.
Think so.
Also joining us today from Western Massachusetts is Alan Dove.
Good to be here, even at this hour.
You usually up at this hour, Alan?
Oh, yeah. Yeah, I've got to take my daughter to school, and,
Yeah.
So
I remember when you worked in the lab, you used to show up at 4 p.m.
No, it wasn't quite that bad. I was usually in by about 10:30 or 11.
I'm just kidding. Also joining us today from North Central Florida, Rich Condit.
Hi, everybody.
Good morning.
Hey, Rich.
Buongiorno. How's the weather down there in Florida?
We got we got 50. I don't know how to describe it except in data. It's 53 degrees Fahrenheit, 19.4 Celsius, and like socked in. There's like clouds everywhere. It's not really clouds. It's like overcast, heavy-duty gray.
I'm so sorry.
Yeah.
I'm not complaining.
Because here it's 4 degrees Celsius.
Ooh.
It was below freezing when I awoke today. But the sky is beautiful. It's blue with no clouds. What do you got, Alan?
I've got 1 Celsius, 33 Fahrenheit, and it's clear blue. Very, very nice skies, but chilly.
I have a great view here from my office. Alan, you probably remember it, right?
Oh, yeah.
I can see the Hudson River and the George Washington Bridge.
Yeah.
And today the Hudson is absolutely calm and sky is blue.
So how cold is it in Michigan?
Yeah, I want to hear this.
It's -3 Celsius and 26 Fahrenheit, and it's bright and sunny. Very nice.
No clouds.
Wispy clouds.
Wispy clouds. Well, there you go. And that'll start your day off. Today's weather.
That'll wake you up.
Today's
you walk
weather in
in
virology.
that, right,
yeah.
Kathy?
Oh, yeah, it's a great one. We have a couple of follow-ups. First one is from Hightal, who writes, Dear TWiV virologists, Vince, Dick, Rich, Alan, and Kathy. You may notice that Dixon is not here. I think it's too early for him. Greeting from Hightal, pronounced Hightal. However, I am only 5 feet 8 inches tall. Now, wait a minute. I'm only 5'7". Are you saying I'm short?
Not tall.
How tall are you, Alan?
5'9".
Kathy?
5'8".
It's okay to ask a woman how tall she is, right?
Yeah.
Because I'm told you never ask them how old they are. And Rich, you're really tall.
I'm 6 feet and shrinking.
Yeah, right. I'm the shortest. 5'7".
Right.
Anyway, the real meaning of my name is ocean wave in Chinese.
Cool.
I'm a big fan of TWiV, and I have listened to every TWiV episode since its first broadcast, but I am always a lurking audience until this email. I also like the weather report at the beginning of each episode. It has become a tradition of TWiV, and thus TWiV can also stand for Today's Weather in Virology. This is great.
Like it.
Yep.
I know there are lots of people out there who lurk. They've been listening since episode 1, and they've never written. But I know you're out there, because I know the download numbers. As an HBV virologist, I greatly appreciate that you guys discussed the recent finding on HBV receptor in the last episode, TWiV 210. The work done by Dr. Wen-Hui Lee's group resolved a long-lasting question for HBV, and it has great potential to set up a game-changing stage in the HBV research field. For over 3 decades, cell culture-based HBV research has been limited to transfection because a manageable and robust in vitro HBV infection system was unavailable. Therefore, such groundbreaking work, although the HBV infectivity in NTCP, that's the receptor reconstituted hepatoma cells, is only about 10% as it has been achieved in primary hepatocytes, gives HBV virologists a big hope to further optimize the infection conditions for the study of HBV biology under natural infection and antivirals as well.
So is this game-changing, or does it level the playing field?
Game-changing. Seminal.
Okay, so it's a paradigm shift.
It's a paradigm shift. The playing field will always be bumpy.
I guess this was the holy grail for the field.
Any other cliches we can bring up here? The paper published by Dr. Bert Semler's group is also fascinating. 5' tyrosyl DNA phosphodiesterase, TDP2, which is shown to serve as an unlinkase for cleaving VPG from the terminus of polio RNA, may also play a role in the HBV life cycle. As you know, HBV genomic DNA, namely relaxed circular DNA or rich Condit rcDNA,
That's it.
also has a protein viral polymerase covalently attached at the 5' end of the minus strand through a tyrosyl DNA phosphodiester bond. Upon infection and also during the viral DNA replication, rcDNA needs to be converted into a perfect circular DNA, covalently closed circular, for viral RNA transcription. So the removal of rcDNA terminal protein is a mandatory step in CCC DNA formation. Our previous work has identified a pol-free rcDNA species in HBV replication cycle, and this deproteinized rcDNA is a functional precursor for CCC DNA. We have also obtained evidence suggesting the removal of pol from rcDNA involves phosphodiesterase activity. Therefore, it is of interest to find out whether HBV utilizes TDP2 to fill the gaps in its viral life cycle. Haha. This is amazing. We didn't get this, guys. We missed it.
Yeah. This is great.
And we had the 2 papers right next to each other.
Right next to each other.
I mean, that's great. It was just a random pick, but
Yeah.
I hope it works.
Yeah.
Yeah, I hope that'd be a great story.
And so polio leads the way for Hep B. Cool. That's really nice. Hopefully we can hear more and more HBV topics in your show. Keep up the fantastic work. Happy holidays. He sent us a Santa HBV, which will be our post art for this time. Really cool. Really nice picture. So the key is, if you write a letter right after the show, like within that week, it becomes follow-up.
Yep.
But if you wait, it may get read months later.
It goes into the queue. Yes.
Heital is a associate professor at Drexel University College of Medicine. Cool, thank you. That's really neat.
Yeah, that's great. I like that association. It's very good.
Really, it makes perfect sense, right?
Mm-hmm.
And that's the protein is a 5' linkage.
I think that's what he said.
The
Yeah.
RNA, yeah, at the 5' end of the minus strand.
Of course, it'll probably turn out to be something completely bizarre that HBV does, but it would be really cool if it uses the same mechanism.
That's neat. Okay, the next one is from Ian, who is writing about TWiV 210 also. I was taken aback by the knee-jerk reaction in TWiV 210 to the Canadian Supreme Court's ruling that it was not a criminal offense for an HIV-infected individual not to disclose his/her status should he/she have an undetectable viral load and be wearing a condom. Sometimes the immediate reaction to such rulings could be interpreted as a proclamation condoning non-disclosure, but this legal statement doesn't mean that moral obligations don't continue to exist. I was glad to hear your opinions evolve quickly as you discussed the topic and explored many important contentious points. Broadly speaking, we have to remember that HIV is spread by consensual adults participating in a sexual act, and at times when neither know their HIV status. That is, individuals acting in a manner that most of us would recognize as ordinary. The mere act of unintentionally transmitting HIV does not make someone a criminal. In fact, it is something that a lot of ordinary law-abiding people unknowingly do. Yet, is there a convincing legal argument for obliging HIV-positive individuals to disclose their status even when they are undergoing treatment and practicing safe sex? Is there an independent moral argument for such an obligation? Now, this individual, so we had said, hey, any lawyers out there, tell us what you think. And this individual got...
This is even better.
Cool. He got trained in virology. Undergraduate at University of Glasgow, worked on interferon regulatory factors. Got a PhD at Cambridge with Tony Minson, worked on simplex, herpes simplex. And he says, in the middle of my virology career, I found myself in law school. But then his interest in virology prevailed, and he ended up doing a postdoc with Max Essex at the Harvard School of Public Health. And that's where he is now. He's working on HIV, HPV, co-infections. I should point out that my comments relate strictly to the question raised by the legal ruling, whether it should be a criminal offense not to disclose one's status should the HIV viral load be undetectable and a condom is used. The willful and malicious transmission of HIV is a separate topic, and I am firmly of the opinion that those individuals should be prosecuted, in which they are, as we discussed last time. Okay.
Then he gives some examples of criminalization of HIV.
That's right, which is... all right, we're not going to read those, but they'll be online.
But he also points out that there's real variation from country to country as to what's criminalized and what isn't. And
from state to state within the U.S.
Right.
That's right. Yeah. Michigan gets an honorable mention here.
Yes.
A biological weapon. Wow.
Yeah.
When he bit his neighbor. Wow. It's worth asking whether the threat of prosecution prevents transmission and increases condom use. Not according to a study comparing the behavior of high-risk individuals in Illinois, where there's a legal requirement to disclose your HIV-infected status to a partner, compared to New York, where there's no legal enforcement of such a provision. Yeah. All right. Then he goes on to tell us some numbers, right? People living with HIV are victims of unwanted transmission. And we have to ask if nothing more than ignorance led to HIV being the only infectious disease to ever have been criminalized. And he gives us some numbers here.
And points out that this is not the only sexually transmitted virus with serious sequelae or infection, for that matter. That's actually something that occurred to me after we discussed this on the last TWiV. I said, well, why isn't it a criminal offense to have sex if you have herpes?
Yeah. Could kill you, right?
Yeah, it could kill you, and it's an incurable virus. So, mortality rate's a lot lower. But
so he says most of these STIs, sexually transmitted infections, can be treated, yet it still begs the question as to why we victimize those with HIV. Right. There's the argument that sex cannot be truly consensual unless participants are apprised of all risk. But where do we draw the line? If people with low risk of transmitting HIV must disclose, does everyone with a known STI have to disclose? Do you have to disclose that you have a cold, just in case your partner has a particular susceptibility? Is this just the beginning of enforced health care through criminal law? Well, if you have a cold, they'll know it, right?
Well, and I was reminded of the... smallpox vaccination cases where there was transmission of the virus from the unprotected scab. And that just, that shouldn't have happened.
But then he goes on to talk about the science, which is strong enough to support the contention that those who are virologically suppressed are not infectious. Swiss government in 2008 went as far as to cause a riot in the HIV prevention field by declaring an HIV-infected person on antiretroviral therapy with completely suppressed viremia is not sexually infectious. It highlights how polarized opinions are at the legislative level regarding the science of HIV transmission and its potential applicability to criminal law. Treatment and prevention of HIV has been the biggest public health achievement of a generation. However, the criminalization of consensual safe sex between an individual with a suppressed viral load and an uninfected partner could radically undermine these achievements, might even have the opposite effect. If the potential for transmission is or remains criminalized, i.e. it's a criminal offense not to disclose your status, those who might be infected may be deterred from testing so that they spare themselves actual knowledge of their positive status.
Right.
This creates a moral hazard where an infected person avoids getting an HIV test and subsequently transmits HIV, yet may not be criminally negligent as she had no prior knowledge of being HIV infected.
Becomes better not to know.
Yeah. This is exactly where all the public health progress in HIV prevention begins to unravel. 20% of HIV infections in the USA are in those who are unaware of their status, but we know that this subpopulation are responsible for half of all new onward transmissions. From a public health perspective, there's no benefit to be gained from the potential, actual, or threat of criminal penalization of the other 80%. We have to ask if society is misdirecting public resources to enforce biased, draconian legislation born out of ignorance about a single virus. Would these funds be better directed to identifying the elusive 20% and possibly prevent half of all new infections. I'm sure this topic will continue to stimulate a lot of heated debate, which is openly welcome, but we have to ensure we're disentangling the moral from the legal question. Should those with an undetectable viral load engaged in consensual safe sex be criminally prosecuted for not disclosing their status? Morally, I would say yes. Legally, no. Yet, like the question as to whether viruses are alive, it may all be a matter of context.
Yeah.
Nice.
Very nice
well
analysis.
put.
Really good.
Beautiful. A lawyer and a virologist.
Yeah.
And I like this segregating the moral from the legal
Right.
obligation.
Yes.
Okay.
But as you know, there are people with no morals.
Right.
Oh, yeah.
Which is why we have laws.
Right.
Yeah. All right. Thank you, Ian, for that. We really appreciate it. And everyone
Yeah,
should
that's very
read the
good.
full letter at twiv.tv/letters.
Yeah.
Maybe most of you don't know that exists, but check it out. All right. On to our papers. Onward. Our first one was actually brought to my attention by Laurel, who wrote, I am a nurse practitioner who sometimes wishes I had gotten a degree in science with emphasis on virology, immunology, but at 50 may be getting too old to pursue. Anyway, I would like to understand more about the function of mRNA in regards to the German scientist who claimed it could be used in production of a more efficient influenza vaccine. And Laurel is from Texas. When I read this, I said, what? And so I searched mRNA influenza, and up came the paper that we are now about to discuss, published in Nature Biotechnology, Protective Efficacy of In Vitro Synthesized Specific mRNA Vaccines Against Influenza A Virus Infection. Who would like to read the names?
I'll read them. Petsch, Schnee, Vogel, Lang, Hoffman, Voss, Schleck, Tess, Cullen, Stitz, and Krumps.
Nice accent.
Nicely done.
Very
Yeah.
good. He could do Finnish too, right?
9.
All right. Now we discuss flu a lot. In
And
fact,
from the names, it might be obvious that these people are all from labs in Germany. Looks like predominantly the Friedrich Loeffler Institute. Is that right?
Mm-hmm.
In Tübingen.
Tübingen, yeah, I like that name.
Tübingen.
Appropriate.
Yeah.
Yes.
Now, as everyone knows, flu is always with us, and we have vaccines, but we need an improvement. And one of the major things we need is to be able to make them more quickly, because new strains arise, and it just takes a long time to make a match vaccine. And we have talked about this before, and one approach is to make vaccines in plants. And Rich and Alan, you did an episode once without me where you talked a lot about this. You can express just the influenza HA gene in plants, and you get virus-like particles, which are immunogenic, and they're being tried in people right now.
Right.
And that can be done in a couple of weeks. You get the sequence from a new strain, and you can make a vaccine in a couple of weeks, as opposed to months with the current egg-based vaccines where you have to make reassortants and test them and grow them in eggs and all this. Now, this paper tries another approach, which is completely new to me, and that is to use what they call the minimal genetic vector mRNA. And, if someone said we're going to use mRNA as a vaccine, from my experience with RNA, where it breaks when you look at it, I would say forget it.
Yeah.
Yeah, that was my first thought, too. Like, how could this work?
But apparently, people are already looking at this in other aspects. It's an FDA-approved thing to use mRNA as a vaccine. It's approved by FDA and European Medicines Agency. And they say that 6 to 8 weeks from the sequence to mRNA, you could have a vaccine. So this is the first time they're trying it for an infectious disease. It's been used for tumor immunology before.
And it strikes me that this is potentially even quicker than any of the other technologies that we've talked about, because all you need is sequence.
Yep.
Right, and you build a clone that can be transcribed. You don't have to express anything.
Yeah.
You don't have to purify anything. Well, I guess you kind of have to purify the RNA. We can get to all the advantages and disadvantages, but it's really pretty amazing. It's also worth pointing out that the idea of DNA-based vaccines has been around for a long time, and people have been working on that, and they've shown some efficacy. But this is my first look at an RNA-based vaccine, and I had the same sort of reaction as you guys did.
Yeah, they say the DNA format still has drawbacks, minimal, insufficient clinical efficacy, residual vector immunogenicity. So mRNA is not replicating; it's just mRNA. It's going to be translated, so it has some advantages. So here they look at it in mice and some other animals, as we'll see. The basic idea is you take an HA gene and they try several. You clone its DNA into a plasmid, and then you do in vitro transcription using T7 RNA polymerase, which Rich talked all about a long time ago in a cool video called Rich Reminisces.
One of my favorite enzymes. I have a long history with T7 RNA polymerase. There's so many people out there who use this enzyme and use the T7-based expression system and know nothing about the phage. We almost ought to do an episode on T7, because it's really a remarkable organism.
I have a sticky here on my computer, one of dozens. It says Studier for TWiV.
Ooh, what a great idea.
That would be really cool.
Yeah, that would be great.
So the idea is you transcribe, you take your plasmid, you linearize it, and you add some T7 RNA polymerase and transcribe it in vitro. You get mRNA, and you make it capped at the 5' end, and it's polyadenylated. So they do this for the flu HA, and basically it looks just like the flu mRNA. They don't make any sequence changes. It's just bona fide cap, 5' non-coding region, 3' non-coding, and poly A. And then you get that. They actually mix it with protamine and then inoculate it intradermally into mice. I don't know if you guys noticed. They say it was complexated with protamine.
Yeah.
Did you notice that?
Yeah.
And what is that about?
I don't know.
Protamine is an arginine-rich nuclear protein, and they give two references, which I was just now trying to look up to figure out if they talk specifically about possibly using other basic sort of nuclear proteins associated with RNAs. But it makes sense. I mean, it's like spermidine and spermine. Yeah. I think protamine is along that line.
So it basically binds
complex,
it, right?
Not complexated.
Yeah.
Right.
The verb got you.
Yeah.
Okay,
complexation.
Sorry.
It's in multiple. No, but that's good to know also. So basically binds RNA and probably protects it.
I think it's like certificated or preventative or one of those.
I'm just wondering if I missed another new word, like reverse genetics or something.
I'll bet you any money they capped this with the vaccinia virus capping enzyme, too.
Yeah, probably.
It's one of these. We use all of these things from all of these different sources. Yep. Viral sources, a lot of them.
Yeah.
And so this complexing with these protamines and the optimization of the structure of this with a 5' cap and the appropriate 5' and 3' untranslated regions and a poly A tail, they think is really important to optimize expression from the message to make this work well.
Right.
And I think the intradermal route of immunization, they don't explore that at all, and they use intradermal in all of these things. And I'm very interested in that and whether other routes of immunization have an effect and why they chose intradermal. I don't know the answer to that, but that interests me.
Yeah, they just seem to say we did it intradermally.
Mm-hmm.
Yeah, yeah, and intradermal is a tough route. It's not easy. That's how you
it's
put
not
the
easy.
It's how you do a couple of other vaccines, and you got to be good. Not everybody can do it, so it would be nice if it worked another way. But you remember the paper we did with skin patches and flu vaccination in mice? Mm-hmm. So it's the same thing. It's basically delivering it to the skin, so that all the antigen-presenting cells.
They may be looking at a delivery system like that.
Yeah.
Yes, I think it's going to turn out that this exactly how you deliver something, in particular with respect to the skin, is going to be important because the skin is such a sentinel organ for looking for pathogens and is loaded with all sorts of antigen processing cells and stuff. So exactly how you deliver something to the skin is going to turn out to be a critical issue in all of this.
Right. It occurred to me it may just be they tried it every other way and this was what worked.
Mm-hmm.
Could be. So they immunize mice with 80 micrograms of mRNA. It's a lot. It's a lot of transcription. And they do 2 at week 0 and week 3, and then they look for antibodies in their serum, in their blood and their serum, and they find them. They either can neutralize infectivity, or they do the HA inhibition assay, which is an inhibition of hemagglutination, which the virus is able to do. So you get good levels comparable to what's considered protective in people, greater than 1 to 40 in the HI analysis. They did it in a couple of different strains of mice. They also get T-cell responses against induced by immunization. They measure that by the production of gamma interferon when they add peptides to T cells from the mice, so you know that they're virus-specific T cells. And then they ask if it protects them. So they immunize. They do this 2 immunization. They challenge the mice with virus, and they are protected. Normally, the mice who are not immunized, they lose weight. Virus replicates in them, but in these immunized mice, fully protected. And that also happens when you give mice the reference vaccine, the standard flu vaccine. So it's pretty cool.
Yeah.
When they took out T cells from these mice, they were still protected. So this is an antibody-based protection. You can transfer protection to other mice by transferring serum. So again, it's antibody-based. And it works against a bunch of different flu strains. They tried H5N1, they tried H1N1, H3N2. And they all work in mice, of course. They work in young and old mice. And in people, of course, the young and the old are particularly susceptible to serious disease. So you want any vaccine to work in them. So it works in young and old mice, but I don't think that is basically predictive of what will happen in people.
Right.
Obviously, it would have to be tested. It seems to be quite long-lived. At least the life of a mouse, 18 to 20 months, they still had protection at 18 months of age after this 2-dose immunization, which is pretty good. Then they asked, can we do it with a single dose? And with a single dose of RNA, the protection isn't as good. You get a little bit of clinical disease. But if you mix it with the HA mRNA with a neuraminidase mRNA in 1 dose, then you get pretty good protection against disease. So they're thinking, maybe we can design a single dose with an HA and an NA component. And finally, they said, could we make a broadly protecting vaccine with this approach? So they make mRNA for the nucleoprotein, one of the internal proteins of the virus. And this induces protection against challenge with a different virus. So they immunize with the H1N1 NP, and then they challenge with an H5N1, and they get protection, which is pretty cool. And this seems to be dependent on T cells, which is what people have always thought these NP antigens induce broadly reactive T cells.
So there must be a lot of literature out there about using NP as a vaccine.
Right. Yeah, people are working on it. They're trying to make broadly protective vaccines, but you don't get the antibody component, and the T cells are what's responsible. You get some cross protection, but it's not great.
Okay.
So there are other approaches that people are taking, which I think are going to be better than NP. There's another one using the M protein, the other internal protein, and they're just not great. And so I think you really need to have antibodies for protection against infection.
Right.
Yeah.
They also check this in ferrets and pigs. And I think the bottom line is that it works in those animals as well. And they do intradermal. And God, I wouldn't want to inoculate a pig intradermally. Oh, man.
Or a ferret.
Or a ferret.
Or a ferret.
I guess. But anyway, it works in them as well. So, you move through a couple of animal models, and then eventually you get into people. So that's basically it. And it's a promising first paper. First time it's been shown to protect, RNA protects against an infectious disease. Now, one thing they say in the discussion, which I thought was really interesting, mRNA has a half-life on the order of days. You put it into these animals, in a couple of days, it's almost all gone. Half of it's gone. It's amazing that it's so effective.
Yeah.
Right.
And so apparently it makes protein right away when you put it in the skin, and that works. And they say also maybe...
I'll bet you it's gobbled up. I'll bet you the RNA itself is gobbled up by antigen-presenting cells that then make the antigen in the antigen-presenting cells.
And that's perfect, right?
And that's perfect.
Yeah. And they also say the RNA probably stimulates innate immunity, which helps get a good adaptive response also.
Right.
Yeah.
Mm-hmm. Yep.
And
unmodified, I would have thought they'd have to modify it to stabilize it or something like that, but no, it's just straight old RNA, capped and polyacylated.
I think the protamine helps a lot.
Yes.
Yeah.
Right.
Yeah, they say that complexation with protamine is necessary for sustained protective immune responses that they see. Complexation.
And I guess they're probably going to take this toward the clinic. A couple of the authors, their affiliation is listed as a company called CureVac in Tübingen. So I think they're trying to make this work.
It's cool. And they talk about rabies. They've done some work on rabies, unpublished data. And so you could imagine this might work for quite a few different vaccines.
Mm-hmm.
I like it, not only because it's interesting and novel, but it strikes me as I said before, almost the quickest possible way from, a new strain of flu to a vaccine, and then it gets around a lot of the issues of vaccine production with contamination with adventitious agents and all of this kind of stuff. This is all biochemistry. There's not even in the production of it. It's like producing a chemical vaccine because it's just RNA. There's no cloning. You don't have to purify the protein away from the organism in which you cloned it or anything else. You're just talking about transcribing a plasmid and making a polymer. That's all. It's amazing.
Yeah.
So you're getting injected along with the mRNA. You're getting a little bit of the template in the enzyme, I presume, right?
Yeah, a little T7 RNA polymerase never hurt anybody. I mean, I'm okay.
Well, and the quantities of that also depends on how they're purifying the mRNA.
Yeah, I would imagine that there's the mRNA is purified, and the T7 RNA polymerase is so good that even in the unfractionated stuff, the amount of contamination with T7 RNA polymerase and plasmid is going to be pretty small. So there would be maybe trace amounts of that in the finished product, but not much.
And depending on how you extract the mRNA, it may be totally inactivated anyway.
Right. Yes, I would imagine it would be.
Well, according to their methods, they don't say that they purify it at all. So,
right.
I mean, if you have to start purifying, then you add complexity to the production sequence, right?
Right.
And I have no sense for how people in the industry view this as a product, right? In vitro transcription, is it okay, or are there issues?
They're probably asking the same questions. They're all scratching their heads, Yeah.
I would expect the FDA would be a lot more keen on this than they are on some of the cellular production systems even.
Absolutely.
Yeah.
I would agree.
A lot fewer components. It's a very, very simple system. You've got very defined portions of it. You'd have to, I don't know, you might have to certify that your T7 is from a proper source and that sort of thing and pure enough. But the stuff that you would have to do, I would think the stuff you would have to do to get this past the regulatory hurdles would be a lot simpler than getting a new cell line or something.
I agree completely.
They make the point that it's unlike a plasmid DNA vaccination, the RNA doesn't have any danger of genomic integration.
Yeah, good point.
And that there's no selective markers like antibiotic-resistant genes in the final vaccine product. So that could make it safer.
Yeah, it really is a minimal vaccine.
Mm-hmm.
Yep.
Why didn't people do this before?
Because everybody says, "Are you crazy?"
Yeah.
Obviously,
you just look at RNA and it disappears. It'll never work.
Yeah, you have to take someone who's willing to try it, right?
I suspect there are probably also some tricks to this that the methods may not go entirely into.
Well, the protamine and the structure of the RNA apparently are critical because they cite some previous studies that have not been very successful, and they talk about the improvements that they've made to make them better. So there's probably been a lot of experimenters that have been discouraged previously by things not working very well.
Yeah, and those things, and maybe intradermal inoculation is crucial, and none of those are obvious.
Right.
Actually, in the introduction, they say provision of ready-to-use clinical-grade product from a pilot facility could be achieved in 6 to 8 weeks from publication. This includes template generation, in vitro transcription, purification, formulation, and quality control. So even though they don't talk about purifying it in the methods that probably would have to be purified if you're going to put it into people, right?
Yeah.
It's got to be a, what do you call?
GMP?
Yeah, thank you.
Yeah.
Okay, so this could be quite revolutionary.
Yeah.
Yeah, I really like it.
Nice.
Yeah, especially if it turns into a general platform.
Yeah,
Right.
could be used for all kinds of stuff.
Yeah.
So good pick, Laurel.
Very good.
Yeah.
There you go. Hopefully that explains it. mRNA.
And we need licensed practical nurses, so don't despair. You know, you're making your contribution both by writing to TWiV and by being an LPN. Keep up the good work.
Well, and she's a nurse practitioner, which is
nurse practitioners. Same
from
thing.
an
Well,
but
not the same thing.
Well, no,
but
not
yes.
the same thing. There's some sensitivity about that, but
Right.
we need those folks.
Yep.
Okay. Next paper. It's a Cell paper. The one-word journal, as Arturo Casadeval says. And this one was picked up by Kathy. A phage tubulin assembles dynamic filaments by an atypical mechanism to center viral DNA within the host cell. Now, Kathy, what grabbed you about this? Do you follow the phage literature, or was there something about this?
I think I actually saw it in The Scientist.
Uh-huh.
And it was while I was eating lunch, and The Scientist had this nice color picture. It was just cool.
Yeah, The Scientist is a good source of TWiVable articles, yeah. Well, I want to tell you that the last author, Joe Poliano, is a good friend of Elio Schechter, my co-host on TWiM.
Aha.
And we have, in fact, talked about the cytoskeleton of bacteria on TWiM. I'll put a link to that episode in the show notes if people want to get a little more background, because Elio is wonderful at explaining it. So what's this about, Kathy?
Well, the authors are from UCSD and UCSF. So Kramer, Erb, Wadding, Montalbano, Zer, Wang, Nguyen, Pham, Agard, and Poliano. And so Poliano is at UCSD and Agard is at UCSF. So just to set the stage, eukaryotic cells that are cells with nuclei have structural proteins that form a cytoskeleton, and that was long thought to be a defining feature of eukaryotic cells. But as has become apparent, prokaryotic cells have cytoskeletons as well. And they're made up of two families of proteins, actins, that form filaments around the circumference of the cell or in through the middle of the cell, and tubulins, which, as they sound, form tubules, and they can be involved in sorting chromosomes during cell division and be involved in cell shape and motility. And so, it's, like I said, only pretty recently, maybe from 2006 onward, have scientists appreciated that the cytoskeleton in prokaryotic cells is there and important, and they're proteins that are semi-similar to to the actins and tubulins. And so there's a lot of prokaryotic actins that are known, and they're quite diverse. And there are a few tubulins, and the most widely conserved one is something called FTSZ. And there's some comparisons in this paper to that.
Would that be Fitzy?
Well, I asked two of my colleagues how it's pronounced, and one of them said, I always hear FTSZ, and I thought it should be Fitzy, but okay.
Do you say bagel or BGL?
Bagel,
yeah,
every time.
Bagel.
Yeah, definitely.
Well, also, if you say Fitzy, then it could be Fitzy staining.
Yeah,
Yes,
fluorescein.
that's a problem.
FITC, which is, okay. So anyway, what these guys did is they searched the genomic database, and that's all the detail we have about it. But in NCBI, there's a database that's full genomes. They just searched that for tubulins, or tubulin-like proteins, and they found some that were within some phage genomes. And then they took and drew a phylogenetic tree from that and found that these things are related to this FTSZ and another prokaryotic tubulin called TWiV, but they're quite different. And then they picked one of them to focus on. But in general, the phages that these all come from are quite large DNA phages, but they infect both gram-positive and gram-negative bacteria. So it's starting to sound kind of universal, at least among the very large genome phages.
Very catchy name.
I didn't know that there were phages this big, or if I knew, I forgot. These are like over 300 kilobases. That's really big.
Yeah.
Yeah.
This is a great name, right? Catchy.
Yeah. So they and just to set the stage just a little bit further, although they've just now identified these tubulins, there's no previous report of a tubulin-based cytoskeleton, and that's what they're about to show, if you remember the title that Vincent read. They decided to name this thing. It comes from a Pseudomonas bacteria, and the phage is called 201 Phe2-1, and they named the protein PHUZ, Phuz, or you could pronounce it fuzzy.
Yeah, I
think
that.
that it's foo myself, but
I think it's probably foo.
Yeah,
but I like fuzzy.
So to study this, the first several figures of the paper, they kind of veer away from virology for a while. But we'll come back to that. They express this protein from as a green fluorescent protein fusion under the control of the arabinose promoter, and that allows them to vary the expression of this tubulin-like protein by varying the amount of arabinose that they give the bacteria, and that becomes important later. And so if they express the protein at low levels in these bacterial cells, they see just sort of a diffuse uniform staining of the from the GFP. But if they put in more arabinose and they induce higher levels of protein, they see it starting to form filaments. And they show movies. These filaments are moving rapidly throughout the cell. And they do some things. They can quantitate how much protein is made. And they show that it polymerizes in vitro in a GTP-dependent manner, and that's relevant because GTP is the nucleotide involved in tubulin polymerization. And then they crystallize it and determine the structure, and they crystallize it using a selenomethionine derivative to aid their crystallization. And they find that it has two domains, like tubulins, an N-terminal domain that contains the nucleotide binding pocket, so that's where the GTP is bound. And then this is for the first time, evidently, in a tubulin homolog, the first time they've seen the structure of the very C-terminal amino acids, and they form an extended tail. So overall, this PHUZ structure has similarity to the other tubulins, but it has some important differences. And one is that it lacks a particular helix called helix 6 that's involved in connecting the two domains, at least in other tubulins. And this suggested that maybe these tubulins have unexpected properties that are going to be different from other tubulins. But they do have this PHUZ has the nucleotide binding pocket and key residues required for it to polymerize. And they show that by making some mutations in the catalytic site in aspartic residues. The mutant tubulin makes these, then they put that mutant PHUZ into cells, and it makes long filaments, so they polymerize, but they don't move. And they show a really cool movie about this, and then another protocol they use is called FRAP, where they look over time, and it's fluorescence recovery after bleaching.
After photobleaching.
Photobleaching, right. And they don't see the recovery, which is telling them that there's something wrong with this protein. It's not recovering. And the filaments also are long, and they seem to sort of chain neighboring cells together. And they say in the figure legend that they're trapped in the septa. So definitely having this mutant protein makes the phage not behave the same.
So it doesn't get that warm fuzzy feeling.
Ooh.
You were just waiting for that, I know.
Yeah, he was working on that one for a while.
No, the way Alan works, they just pop into his head.
They do just pop into my head. That one did pop into my head a minute ago, but
So they're able to look at this filament structure in the crystals that they use in the crystallography, and then they show some pictures that show that the crystals that form, or the filaments that form in the crystals, are like the ones that they see in solution, and then that they then prepare for electron microscopy. So that's a nice correlation. And then getting back to the C-terminal tail, they say that the C-terminal tail they show that it's really important for polymerization. And this seems to be unique to this tubulin so far. They make a truncation mutant where they lop off a big portion of the tail, and they make 2-point mutants. And those mutants just don't polymerize. They show that in vitro. And then when they put these mutant fusy expression plasmids back into the Pseudomonas, they have to use really, really high levels of the arabinose to get really high levels of the protein expressed, and then they kind of start to see the filaments forming. So now they've characterized it, and now we're kind of going to get back to the virology and see what fusy is during the infection. So they use this really cool assay. So first of all, they're going to grow the cells on an agarose pad. And in the pad, they put DNase-1, which will digest unprotected DNA, and they put DAPI, which will stain DNA a fluorescent blue color. They put in a membrane dye that will stain cell debris really intensely, but hardly stains intact cells at all. And so then they have Pseudomonas that has their GFP expression plasmid, GFP fusion in it, and they grow those cells on this agarose pad. And then they add phage, in which the fusy won't be labeled. And what I forgot to say is they're growing the cells in a relatively low amount of arabinose, such that the fusy alone won't polymerize. It'll be that same diffuse staining that you see when it's expressed at low levels. And so when you add the phage, if you get polymerization, it's either because the phage is now providing more fusy or some factor that enables it to assemble. And I'm not sure they ever really completely address that it's more fusy and not a factor. But
right, I agree, yeah.
So anyway, they do this, and so they do this a couple different ways, and they look at things. But so the first thing they do is they show us is this movie, and I say that the money figure of this paper is figure 6A, because this is just stills from this thing. So now I'm going to explain why they put all those different things in the agarose pad. What they expect is that while the phage is replicating, the DNA will start to get encapsulated, and when the cells lyse, the cellular DNA will spill all out and get digested by the DNase, but the phage DNA will not. And then you'll be able to tell that the cell's lysed because you get this bright red staining from all the cell debris. And so they show that, and then what they also see are these blue dots that are presumably the encapsulated phage DNA that's come out of the cell. So it's just this lovely assay. And they show a movie, but the movie doesn't really have the blue staining. So I just thought that assay was...
It's nice, yeah.
It's amazing.
Yeah. So then they do another kind of experiment where they do the same kind of thing, but they stop the cells before they lyse and fix them and look at the GFP staining, and they stain with DAPI. And so they see this really weird thing where in the middle of the cell they see this big bulge, and associated with that is a big bunch of blue staining DNA, and they call it a nucleoid. And the amazing thing is they quantitate how many cells have this very close to the center. And in 98% of the cells, it's within 10% of the middle. So this mechanism is somehow getting this DNA and this clump right to the very middle of the cell. One thing I wondered about is they don't ever talk about seeing the cellular DNA.
Yeah, I was wondering the same thing. The presumption is that that's phage DNA, right?
Well, because they have the DNase in there, right?
Well, not in intact cells.
Right.
And they do say that when they digest some of the that some of the stain in the clump goes away. So presumably some of the cellular DNA. I just wonder if it's all of it, or does it get pushed into the center too?
Yeah.
Well, there are some phage that actually cause the cellular DNA to be degraded.
Yeah,
I don't know if that's the case in this.
Yeah, this one does.
It's, T4 phage, if I recall correctly, it infects and degrades the host DNA and uses those nucleotides for making its own DNA.
Yeah, so figure 7, they say the phage infects. The host chromosome is degraded.
Okay.
So I think by the time that they've done these photos, it's probably the case that it's been degraded. Yeah.
Right. So
because it's 90 minutes post-infection, so that's like a long time. Right?
Mm-hmm.
Phage, yeah. They do optical sections, and they show that in this center of the cell, they get these blue dots, and they form this really beautiful rosette structure. And if you look at the movie online, it rotates it, and you get to see that. It's worth looking at. Okay, so then they go back and they look at that mutant that makes the immobile filaments. And when they put that into this assay, these nucleoids, which is what they're calling these DNA masses, they're not at the center of the cell. And they also see that virus yield is reduced. And they say that instead of getting about 15 phages per cell, they only get about 7 phages per cell in the yield.
And that mutant is a mutant that cannot hydrolyze the GTP. Is that correct?
I think that's the case by analogy with, it's in the active site. And so it's the key catalytic aspartic residues.
So I was, just to make sure I'm clear on this, the role of the GTP is not in polymerization, but in motility of the filaments once they're assembled, right?
I think that's the conclusion I came to also, yes, because okay. So, yeah. Anyway, they call these filaments a positioning spindle, and that sort of makes it analogous to microtubule-based spindles that are in eukaryotic cells that position chromosomes. And then they talk about how it does this by assembling and disassembling, and then that there's always one continuous filament that they see throughout until the cell lyses. But there's a lot of disassembly, and then presumably when it's reassembling, that's somehow pushing the DNA into the center. That's how I interpreted it.
Right.
And that when you have the mutant, it can't do that assembly and disassembly, and the filaments are immobile, then you don't push the DNA to the center. So they say that the bulge forms even when the DNA is mispositioned. Mm-hmm. So the bulge itself is not just from the DNA being there. And so they hypothesize that maybe it's where the capsid proteins are and where the assembly has to take place. So it was nice because there are analogies to what we know about for DNA viruses assembling or replicating their DNA and assembling in factories, and RNA viruses doing that, and they make that comparison as well. So I thought that was really cool.
Yeah.
And then one other thing for listeners who might not ordinarily read scientific papers, these authors have what's called a note added in proof. So you submit the paper, it gets accepted, it gets typeset, and then you get to look at it again one last time. And in this case, a paper came out in another journal by some other authors identifying another phage-encoded tubulin structure. And so just to say, okay, we now have, we're aware of this, and there's this other group that did this, but instead of doing this kind of pushing DNA to the center of the cell mechanism, the one identified by the other group, which is Olivia et al., is that it seems to be involved in plasmid segregation during lysogeny, which is a different aspect of a phage life cycle. So it's nice because they acknowledge that someone else has worked on this too and found something, and we can, you can go check that out.
So basically, the phage infects this bacterium, and its DNA is positioned in the middle of the cell by these fuzzy filaments, right? That's the general idea. So why does it have to be in the middle?
Who knows? Maybe that's just the way the filament mechanism can do it most easiestly.
Right.
There's a lot of that would make sense to me. That if you grow these filaments and they're somehow contacting the inner surface of the cell, that the sort of dynamically the most logical thing to happen is that it would be aggregated in the center. I think the important thing is that, or what they speculate on a lot, is that the major function of this is to concentrate the DNA in one place.
Right. I think the center is just the easiest point to find.
Mm-hmm.
Well, because in other virus-infected cells, like the replication centers are all over the cytoplasm, for example.
Right.
They don't have to be in the center of a cell.
Right, but in this case, it may be, rather than having replication centers in multiple spots, this tubulin mechanism was a convenient way to just put everything in one place. And if you're going to do that, and you're doing it with these polymerizing tubes, you could easily imagine a mechanism where it's based on the tension being equal from both ends, for example.
Yeah, so that's the way these are. These are bacilli, so the poles of the cell help to orient it, right?
Right.
And if you have equal force from both poles, then it ends up in the middle. It's a similar thing. We have a guy doing S-Pombi cell division here, and it's a similar issue. You know, the ends are used as reference points for cell division, and it happens in the middle. And if you muck with the machinery, you get the dividing plates, those things in the middle before the cell's divided at one end or the other by mistake. You know, when you alter the machinery. It's pretty neat. This is really a nice paper.
For some reason, and I don't know why, I really like the EMs of the filaments. Mm-hmm. Somehow those grab me. I think they're really cool looking.
It's always cool to see EMs.
Mm-hmm.
So you mean like figure 3C?
Yeah.
Yeah, that's pretty neat.
Yeah.
So Kathy, why is this important to study?
Because it tells us about how things move around in cells. It's important. It's important cell biology information. It's important because it is.
I think it's also important from an evolutionary standpoint.
Yeah.
Okay, the part of the lead-in here was that we always thought that cytoskeletons were unique to eukaryotic cells, and now it's apparent that that's not the case, and they're throughout nature.
Yeah, and bacteria obviously have, as a group, have enormous medical importance. And we'd like to know more about how their cytoskeletons work. And here you have a model system. You've got this phage that is exploiting its own tubulin mechanism to accomplish something in the cell, and you could maybe use it as a way of getting at what else is going on with the cytoskeleton in there.
Yeah, and as you know, we always need more antibiotic targets, right? So this could be another one. Yeah.
If I understood it correctly, they did build a phage that had a mutation in this thing, and I think it was the GTP hydrolysis or binding mutation, and that had about a 50% reduction in yield, if I read that correctly.
Yeah.
Just doesn't necessarily seem like a lot, but if you throw that thing out in the biosphere, it's going to fall flat on its face.
Yep.
Yeah.
Yep. Go ahead.
That terminal structure they did say is found in some other prokaryotic tubulins. So maybe not even just phage tubulins, but this is something that some other bacterial cells are exploiting as well.
Yeah.
Right,
So-
and the phage presumably got this from somewhere.
Right.
I don't know. It could have evolved independently in the phage, but it seems more likely, especially with the genome size of this thing, that it picked it up from some bacterium that it infected.
Or it depends where the virus came from. I mean, if it may
Yes.
have initially evolved as a cell or an offshoot of a cell and then just lost genes, and this was one of the ones that kept. Who
Right.
knows? When we did a TWiV on this with Elio Schechter, you have to go listen to this TWiV 28. The title is Not Unorganized Bags of Enzymes, because Elio said back in the day we used to think bacteria were bags of enzymes with no organization, and now this is really changing. They clearly have a cytoskeleton. Many of the things we thought about bacteria, it's the same for all of science. We establish these dogmas, and then they all change, which is great. There's really no truth in the end, right? But there is. We just can't figure it out.
Well, you get provisional truths that can be extremely useful.
Yeah, Yeah.
And then you as you try to extend them, you find out, hey, there's more to the story.
Yeah, right.
Well, the fun part about that is that you have to keep restructuring your mind as things change. And I think that that's one of the most fun challenges to all of this.
Right.
Oop, wrong again.
There are many examples in science where people have proclaimed, a problem has been solved, and it's never the case, right? There's always something else. Yeah, remember the Surgeon General said in the 60s that infectious diseases were over. We'd conquer them with antibiotics. All right. Anyway, that's cool. Thank you, Kathy. That was really nice.
That's great.
Very nice. I like that very much.
Good pick.
All right, let's do a couple of emails. Rich, can you do Christine's?
Sure. Christine writes, hello. I'm having some problems understanding how the triangulation number of icosahedron or icosahedral-shaped capsids is derived. What does it represent exactly? Trying to find answers on the internet only got me formulas to calculate T, but not a description in plain English of what it represents. I thought I understood with the lecture material of Professor Racaniello on iTunes U, but was not able to put it in words or apply it to other icosahedrons. Thanks for your help, Christine. Go for it, Vincent. You're better at this than I am.
Basically, an icosahedron is made up of triangular faces, right? And the T number is just the number of facets on each face, how much you can divide each triangular face. And I will post a picture from our textbook about this, which shows it and shows two different examples of a icosahedron with a T equals 1, where there's just one triangular face, there are no subunits or facets, and then one with a T equals 3. So in the simplest terms, that's how I remember it. It's the number of facets on a triangular face. So if you envision an icosahedron, as a geometric shape, I think you'll get it that way.
So then you say that the minimum number of subunits required, and I guess that means for an icosahedron, is
60, yeah.
So then the total number of subunits is 60 times the T number.
Right.
Right? So if it was a triangulation number of 1, then there'd be 60 subunits. But if T equals 3, then there'd be 180.
Yeah, that's right. Right. I'll put a picture up, Christine. That'll really help. All right, let me take the next one here. This is from Anonymous. Hey, TWiV team. Now, I'm going to do a little paraphrasing here for reasons that will be obvious. So this individual is a graduate student who has finished the second year, long-time listener of TWiV, does some teaching and listens to our podcasts. And she likes our podcasts because they can help expose her students to different areas of microbiology in a fun, low-stress way. And she particularly likes our podcasts for grad school when it gets tough. Now, she has been asked to leave her lab, and that's what this email is about. And the quote, the reason she was given, here she writes, the reasons given were ones I'd heard once earlier in my career. You're so smart and so good at reading papers and synthesizing information and deeply understanding the biology, but your bench work is so atrocious that I think you are a danger to my lab and I need you to leave. All right, so she gives a lot of background, which I will skip, and whether I put it on the website depends on the individual who I will ask. But let's cut to the chase here. Being asked to leave her lab a year into her thesis work, she could choose a new lab, but she's being advised away from bench work by her PI. And they've promised to actively try to prevent labs that have any project with live human pathogens from taking her. This largely restricts her from all the work she came to graduate school hoping to do. I've been advised to try bioinformatics lab. However, I have no training in bioinformatics, and while I'm smart and learn quickly, I would realistically require another full year of coursework to be successful. I find it hard to believe that anyone in bioinformatics would be thrilled to get a third-year student knocking on the door because they failed at what they were actually interested in. If I choose not to find a new lab or cannot find one by November 30th, which of course has passed, I will spend my winter quarter writing a thesis and will graduate with a master's degree. For what it's worth, I'm not completely incapable in other aspects of science. I have a published review, and even the PI who is firing me says I excel at writing, reading papers, synthesizing information, and designing experiments. Even before this happened, I was investigating my options in science communication or teaching or maybe policy or consulting. So as I see it here, my options, I can try to get into a lab that would require bench work against the wishes of my current PI. This seems unlikely to work, but it's possible. 2, I can fight to try and find a bioinformatics lab willing to take me and then spend 5 years working on my PhD. 3, I can leave with a master's and start now trying to get the same types of job after I'd got my PhD, just without the advantage of a few more years to prepare myself for the job market and with the less impressive letters after my name. So I'm investigating options 2 and 3, and 1 to a lesser degree following the advice of a professor I rotated with who sadly can't take me due to budget constraints. My question to you is this: given that I don't want a research career anymore and my options for new labs are limited, do you think the uphill battle I'd be facing to even earn the right to try to finish the PhD is worth the effort? What do you say, guys? Any insight as to what to do with a student who is supposedly brilliant but useless on a practical level? So I know that the decision has probably been made, but it's worth discussing a bit because this happens in science. So what do you think, Alan?
Well, my take on reading this, and there's a lot of detail given in the specifics, which kind of illuminated this for me. But I don't know if we're posting that on the website because the specifics of the case would make this person identifiable. But a lot of the incidents that she describes from the lab, I was thinking this sounds like somebody who has some kind of actual attention problem. And I'm not qualified to diagnose anything. I'm not certified to diagnose anything, but I happen to live with a psychiatrist, and I know that there are conditions, both psychiatric and neurological, that can cause this type of problem where people are, smart and bright and do fine in a lot of, in abstract reasoning and all those sorts of things, but they, walk off and leave the keys in the door and forget things all over the place and forget they left the stove burner on or what have you. So my central piece of advice to this person is before you make any more big life decisions, go and see a psychiatrist. And specifically, psychiatrist, your insurance company will probably try and shunt you toward a therapist or something. But I think in this case, you need somebody with medical training who is qualified to evaluate your symptoms, as I would see them, and possibly prescribe something. Because it may be that you have something that's very treatable that is causing this to happen. It may also be that you're just not cut out for science. You need to keep that possibility open. This might not be a psychiatric disease. But if it is a psychiatric disease and you don't get it treated and you switch to some other career and you decide to become, I don't know, a science writer, it's going to be an even bigger problem there. This is not going to get better if it turns out that there is an actual treatable condition underlying it. So please, that would be the first thing to do. I
think that's great because it could, as you say, it could be treatable. And then maybe a lab career could work under those conditions.
Yeah, and there are. My wife has stories about patients who, they just couldn't get together, and then they get the appropriate treatment, and it turns out, well, yeah, this was a biochemical problem with their brain, and then they go on to do just fine. Yeah, so if that's what's at issue here, then you should focus on fixing that. But as I say, that may not turn out to be the answer.
Of course.
So just go to a psychiatrist and give an honest history and answer their questions and, proceed from there.
Yeah. What do you think,
Yeah,
Rich?
I went through this in graduate school. I came to a point where at one point the stress was overwhelming me, and I went and saw, in my particular case, I had some very specific symptoms that ultimately took me to a neurologist. Okay, but I went to this person and I said, I think I'm probably just crazy, but I want to make sure first that the machine is actually okay. And he said, he examined me, fascinating. Neurological exams are fascinating. He examined me and he says, "You're right. You're crazy. The machine is fine." He says, most type A's like us at some point in their career reach the end of their rope, and he says, you just found yours. And it's been a lifelong issue since then to deal with that kind of stuff. This stuff needs to be dealt with, and then you can get on with the job at hand. I agree with that. And beyond that, I think it's also, there's a lot in this letter with people pointing out what her strengths and weaknesses are and advising her in a certain direction. And this idea that you're really good at some stuff and some stuff you don't seem to be good at, once we understand whether or not the machine's functioning correctly, okay? I think that's an important point. But, if that all turns out to be okay, she's got some from a number of different sources, people telling her what her strengths are, and I think that's worth listening to, okay? Because, and I also think she points out that there's one person, at least that she's talked to, who's been very understanding and a really good mentor. Obviously, she has some history with some other people in most, if not all, academic environments. There's going to be at least a few people who will really listen to what your issues are and try and help you find your path rather than trying to push you one way or another. Try and understand who you are and try and help you find your path. And there's no glory in a PhD. You don't have to have a PhD.
Hear, hear.
Okay? It's not a big deal, okay? And there's plenty of stuff where you can have a perfectly satisfying career and have a great life where PhD is really overkill, all right? So take the time to make sure that the machine is operating well. Find somebody who will really listen, who you can really talk to, and then, take the time to follow your strengths, okay? And, the PhD should not be a goal in itself. Try and find, where your passion is, and a lot of that is what you're really good at, and just let that happen.
Yeah.
Yep, I agree. I would say that, go and listen to John Udell's TWiV, and, science is really hard now. And if you can't really work in a lab, you can't do it. It's not going to work. So if it turns out that, as Alan suggested, you can't be treated, you can't work in a laboratory in the way that you've described to us. So I would say that's not something for you to do wet research, wet bench research. But as Rich says, find something else. You could. Bioinformatics is great. It doesn't matter if it takes another 5 years. You don't have to get a PhD. But if you would like to do that, just find what you like to do. But I would really strongly suggest that stay away from the bench if you have problems doing wet bench research that can't be fixed by treatment.
Yeah, I was just going to say the same thing about if it takes another year to get your bioinformatics courses and you can get turned on to that with the interest in the microbiome, fine. But I had my first technician. I had started a PhD, and then she moved to Georgia where I was, and she became a technician. And then she moved with her husband, was a great technician various places, and eventually, I think she was probably in her 40s, she went back and got her PhD. And then, a couple years later, she got a master's in library science, and she's working in a high school library, and she loves it.
Yeah, sure.
That's great.
It's like people used to write to Dear Abby, well, if I go to medical school, I'm 40, then I'm going to be 44 when I get out. And Abby would always say, yeah, you'll be 44, and if you don't go to medical school, you'll just not have the MD degree.
You'll still be 44.
You'll still be 44, yeah.
Yeah.
So,
right.
There's lots of time to explore different things. And, if bench science isn't going to do it, there's, as Rich was saying, there's lots of things. The PhD isn't everything, and lots of things that you might find that you like.
Yeah, so see a shrink and then evaluate your options.
So she, I don't remember actually what the gender is here. She had written in an addendum, don't be obliged to talk about this. I do have mentors here. I'm just looking for some advice outside my particular little bubble and thought you guys might have some insightful conversations. So I hope that's useful, and good luck. But don't push it either. It doesn't have to happen right now.
Mm-hmm.
Yeah.
All right, Kathy, can you take Charles?
Charles writes, Dear TWiV team, during episode 198, you talked about the eternal competition between viruses and their hosts, the so-called Red Queen's race. It occurred to me that the character of the race must have changed when animals evolved sufficient intelligence to know to avoid sick and therefore infectious neighbors. Would this have put selective pressure on viruses to either not produce visible symptoms of disease or to take a long time about it in order to provide a window in which their host could infect others? For humanity, a much more important change to the nature of the race must have happened in the last 200 years. Our increasing intelligence has got us to the point where, unique among all the species on this planet, we are actually aware of viruses' existence, and we use our intelligence to develop treatments against them. From this perspective, it might not be too much of an exaggeration to regard virologists, with a capital V, as an evolved response to the problem of viruses. We must hope that viruses don't retaliate by selectively targeting virology.
This is really great.
So, more seriously, given the extra effort that humans are making to defeat viruses that infect us and those species dear to us, are there any signs that these viruses are now evolving faster than viruses that aren't in our sights? Please keep up the great work with all your podcasts. I now listen to TWiV, TWIM, and TWIP, even though I've had to increase the number of times a week I go to the gym to find time to listen to them all. Yours ever gratefully, Charles.
Yeah, you're getting healthy there. It's good.
Yeah, that's great. We're having lots of beneficial effects here.
I think this is fascinating. I've often mused on what the human intelligence, what the impact is going to be on our evolution. It's too soon to know.
It's going to wipe us out.
Yeah, possibly.
I mean, dinosaurs were the dumbest, and they lived 140 million years, right?
But the spin out to viruses attacking virologists, I think, is just wonderful.
I love that.
Good.
Yeah. So to sort of address the question more seriously, are there any signs that viruses are now evolving faster than viruses that we don't know about? And I would have to say probably not. That we know something about mutation rates of various types of viruses, and just because we may be developing antivirals or vaccines or something like that, it's not really changing how viruses evolve.
Yeah, they've always had the capability of evolving very, very rapidly. And
you could argue that in the bacterial world, our use of antibiotics has changed the landscape, right? Because all these resistance genes, which were in the soil before, we've now introduced into people by doing this.
Yeah.
And I don't think you see anything like that scale with viruses, at least not yet. We don't have enough antivirals to get at that point, but it could be. But I agree. I don't think we've made any dent in what viruses do.
And the scale, we think of ourselves as this hugely dominant species on the planet, and in some ways we are. We're changing the climate and things like that, and we're fishing out the oceans. But from a microbes perspective, most viruses and bacteria, we're not even a surface to them.
Right.
There's just huge diversity of microbiology going on in the oceans, which are 2/3 of the Earth's surface, and that just has no direct relation to us whatsoever. So the stuff that we think about in medical microbiology is a really, really tiny sliver of the whole field.
Yep. Hey, Rich, I think you ought to take the next one.
Yeah, my buddy Neva.
Mm-hmm.
Neva writes, hi, fellas, and Kathy. Hi, fellas. Thought you would enjoy this piece on the future of biodesign. The handle Captain Capsid is pretty fun. Oh, my. Best to you all, Neva from Buda.
Hey, it's hi, fellas. She's like imitating you, Rich.
Ah, could be, yeah.
She knew you were in Buda.
Yeah. So she gives a link to an article that basically talks about the ramifications of our understanding of the genome. Okay, and it starts with an apparently sci-fi scenario about designing a specific virus that can kill just the president based on understanding what the president's susceptibilities are based on the president's genome, and talks about all sorts of intrigue where in political circles people are interested in the genome sequences of various political potentates and et cetera. It's quite an amusing article.
I like this, one of the protagonists here, the winner of this contest, right, is a 20-year-old Columbia University undergrad with a knack for virology.
Is that what students?
I wonder if he took my course. It's just fictitious, but we're not even close to this yet, right?
No. To point out that Navy stewards gather bed sheets, drinking glasses, and other objects that the president has touched so that they can be later sanitized or destroyed. But the Secret Service would neither confirm nor deny this.
Right.
I can neither confirm nor deny. Sorry, I just had to.
Right. And then also there's something that evidently was in WikiLeaks that perhaps DNA samples are being collected from foreign heads of state.
Yeah.
So, it has occurred to people. It's not all fictitious. So it's an interesting article.
I don't know what you can do with the sequence, though, because it's just we can't do anything of that sort yet, and Obama will be done in 4 years, and every other leader will be gone. And so by the time this comes to some fruition, it's going to be a long time.
Yeah, and this notion of a personalized bioweapon has been kicking around for a while, and I just the more we learn about how viruses behave, the less likely this scenario seems.
We just got finished saying that we don't understand anything, right?
Right.
You know, that, it's changing all the time. And so the notion that you're going to be able to really figure out something nasty and highly specifically nasty from the genetic code is pretty far-fetched. However, the point is well taken that the ultimate ramifications of having all of this genome sequence are both complex and interesting.
Yep.
And unknown.
It's a good article.
Yeah.
Check it out. Alan.
You want me to take the next one?
Did you read one?
I didn't read one
No,
yet.
go ahead, Barbara. Yeah, please.
Okay, so Barbara writes, Dear Sirs, my name is Barbara. I'm a student from University of Fernando Pessoa in Porto, Portugal. First of all, I would like to apologize for my English. It's not perfect. It is far, far better than my Portuguese, though. I'm a reader and listener from your website and podcasts, which I appreciate a lot. I'm now on the last year of my course in pharmaceutical sciences, and I'm starting my final project. My project theme is viruses as machines in pharmaceutical sciences. When I thought about it, I've had some ideas, such as vaccines, gene therapy, viruses and bacteria, biotechnology. For example, is it possible to introduce some genomic material of a virus in food that with metabolic processes would be introduced into our genome in order to fight some diseases? In synthesis, how could a virus be more useful in developing new drugs? The purpose of this email is to ask you if you could give me some more ideas so I could develop my project correctly, and if possible, send me some reading material about this theme and these sub-themes. Thank you very much. Yours truly, Barbara.
There's some good ideas there on your own.
Yeah, she's already off to a great start. Just follow those. There's nothing wrong with anything she's got there.
And the name of the field for the food modification for therapeutic uses is nutraceuticals, which is an unfortunate term, but it's nonetheless become an area of some industry research.
So you could take the mRNA paper today and use that, for example.
Yeah.
I have a feeling Barbara is where our friend is in Portugal there. Fernando Pessoa sounds familiar. You know, our friend whose name is now escaping me, and which is really not good.
Is it a Ricardo? Or
Ricardo. He writes us all the time.
Regularly, yes.
So maybe she knows him. All right, let's do one more from Robin, who writes, in the introduction, you say the kind that make you sick. We should all acknowledge that all that retroviruses do for us. They or their remnants in the human genome literally keep us going. Metabolic processes essential to us depend on them. Even the continued existence of all placental mammals depends on the syncytian gene co-opted from some ancient friend to make the syncytiotrophoblasts of our placentas. While the disruptive effects of the viruses are more readily recognized, their beneficial effects working quietly in the background are harder to detect. In a very real sense, viruses are us. Perhaps the introduction should be the kinds that make you and the kinds that make you sick. Love your podcast. Yes, we have talked about this before, Robin, and I know the title isn't perfect, but I really like it.
Yeah, I've come around on it. Initially, I thought, well, it's not just the kind that make you sick, but it really works.
Well, one of your initial motivations, Vincent, as I recall, was to differentiate this from computer viruses.
Exactly, right? Yeah. So it was a Saturday. I had just finished editing the first episode of TWiV, right? And I'm ready to post it. And so I needed a little intro. What am I going to do? And I said, there's so many computer virus podcasts out there, and that's why I did this. And it just came to my head, and then I think I went and asked my daughter, who was probably 9 at the time. I said, what do you think of this title? She said, yeah, it's good.
Yeah,
so
it sounds so good.
But it does have and even Kathy Spindler wrote before you came on the show that you liked it.
Yeah, but it was at the Bozeman ASV, where it was Marilyn Rusink talking about virus don't make you sick. And I remember you talked about it there that, Yeah,
there are plenty and lots of examples, and who knows, maybe the majority are beneficial. So I just like the tagline. I'm sorry.
And we've played with it in the title, too. Didn't we have an episode called The Kind That Make You Better?
Yeah, exactly. So.
And I like Viruses Are Us. That's good.
That's good.
Yes.
I like it very much. Thank you. Thank you, Robin. All right. Let us do some picks of the week. Rich Condit.
Okay, I have something that was sent to me by my son-in-law, Joey. I think he's the one who sent it to me. At any rate, it's the Global Shark Tracker by Oshreach is the organization that does this. And this is an organization that is studying sharks. And they capture, I'm sure it's traumatic for a period of time for these sharks, which doesn't make me real happy, but they capture these sharks briefly and take a bunch of measurements and stuff, and then attach a satellite tracking device to their fins, and they can keep track of them for a long period of time. And they got about 20 of these guys, all with nice names. I think, did I see a Vincent here? Maybe not. No, there wasn't one. Okay. And
I see Mary Lee is suited for you.
They can track their movements and stuff like that. There's one that I'm looking at here. I think it's is it Mary Lee?
Mary Lee is down off Georgia right now.
Right, and if you have a look at her, it gives you all the data. Okay, so she is I can't pronounce it. Carcharodon.
Wait, let me get back to species. Carcharodon carcharias?
Very good.
Wow, 16 feet long.
16 feet long, weighs almost 3,500 pounds. And she was tagged in September of 2012 in Cape Cod, and she really, in the first little while, tooled quite quickly down to Savannah.
Oh, you could see her whole path.
She's been hanging around the coast of Georgia and South Carolina now for quite a while.
And by the way, Carcharodon carcharias, for those of us who don't study sharks, that is a great white.
Right.
Okay.
I think a lot of these are great whites. So there's two now that are off the coast of the U.S. Most of them are off the coast of the southern tip of Africa and in the Indian Ocean. They got about 20 here. So you can pick your shark or several and follow them around for a while.
Hey, listen, we got to do this with viruses.
You're going to put a satellite tracker on a virus?
I want to see where they go.
Yeah, okay.
This is out of names.
This is just great.
My first thought is that when this opened up, there's all these sharks around the southern part of Africa, which is where all those sailboats in the Vendée Globe are.
Nice.
Did the sharks cause the sailboats to be there?
Could be.
Nice. That's really great. Good find. Alan, what do you have?
I have a project. This is from the Smithsonian National Museum of Natural History, and it is the Field Book Project. As somebody who trained in bench research, I've always been kind of fascinated by field science and people going out and doing interesting things out in the world, and that's their science. So this project is collecting historically important field notebooks from different expeditions, going way back. I think they just recently got started, so there's not a whole lot here yet. But they have a Flickr page where they're uploading scans of these things, and just some really neat bits of history.
Cool.
Yeah, it looks really neat.
Yeah, very cool.
So they don't want your lab notebook, right, Alan?
No, that would not be a field notebook or historical.
It's really cool.
Nice.
Yeah.
Kathy, what do you have?
I have an article that was in the New York Times on Tuesday in the Science Times by Carl Zimmer, and the title is "How to Manipulate an Army of Zombies." And it most talks about various insects and fungi and things like that, how parasites control the brains of their hosts. But they do mention one virus, and it was just such a cool factoid that I had to mention this, that talking about baculoviruses, which is found on cabbage, and so the cabbage in a serving of coleslaw carries 100 million baculoviruses.
Yep.
Right.
Yep.
Baculoviruses won't hurt you. But anyway, I thought it was a really interesting article to read these various possibilities. And then the closest thing, which maybe you've talked about on TWiV, to something like a virus, I thought was the parasite toxoplasma that could potentially be doing things to human brains.
Right. I did a little talk about virology this week at a local restaurant. It's part of a Columbia series. Columbia professors, you sit in a restaurant and people eat dinner and they listen to you and then they ask questions. One guy asked me, are there viruses that go in your brain and make you a zombie and attack other people and give them your infection? So after we came back from Brazil, Rich, this fellow George Rohrman gave me Baculovirus Molecular Biology, a book that he wrote.
Right.
And I was reading it, and that's where 100 square centimeters of cabbage. So he did a study. In one study, it was found that cabbage purchased from 5 different supermarkets in the Washington, D.C. area were all contaminated with baculoviruses, such that each serving would contain up to 10 to the 8th polyhedra.
Yeah, I wouldn't even call it contamination.
Yeah.
You know, contamination makes it sound like they shouldn't be there. Okay? The
Right.
baculoviruses have every right to be on the cabbage.
Yeah.
And they're not doing you any. I mean, it's
I
good
would
protein.
call it protein enrichment.
Yeah, exactly.
Indeed. No
But we get these questions about, do microorganisms? We had a letter about it today. Do microorganisms, modify behavior of things that they infect to their advantage? And this article is a nice, rich resource of examples of just that.
Sure.
Yes, very good.
Well, my students who were there at this thing mentioned rabies and making dogs aggressive so they bite other animals and transmit infection. So there are a couple of examples.
This thing you're doing, is it like cafe science or something?
Yeah, it was called
like
Cafe Columbia. It's it's science and everything else that Columbia professors do.
Okay.
And do they serve Colombian coffee?
No. Well, I might. I didn't know. But the cool thing was they had I went to the restaurant. It was on Broadway in 102nd. And outside the restaurant, they usually have a chalkboard with all the menu on it, right?
Uh-huh.
So for that night, which was Monday night, they had Columbia cafe, virologist Vincent Racaniello tonight. And then on the side, they had, it's fish night, moule frite or fried sole. So I shared the menu. It was great. I really liked that. And I put a picture of that on the Facebook page, facebook.com/thisweekinvirology. And one of our fans who wrote, did you talk about marine viruses? It was good. My pick is a site I just found today. Science Sculpture by Julian Voss Andrei. And this fellow does science sculpture, and on his front page you can see an antibody. He made this cool metal sculpture of an antibody, and he's got a bunch of others in there.
He's very prolific.
Yes.
He's got all kinds of stuff. It's really very cool.
It's very cool. The reason I found this is so cool. I was asked to review a paper today in a journal called Leonardo. It's an MIT journal. It's about the intersection of science and art. I was asked to review a paper by an artist who does microbial stuff. It's probably the most fun paper I'm ever going to review. Anyway, and I found this in the reference. So that's that. We have a listener pick of the week from Stephen. I ran across this site and thought it would make a good TWiV listener pick. It sounded familiar, and I thought it had already been mentioned on TWiV, but I did not find any mention of it, and the site has a lot of interesting animations. This is a cool... it's a TV site, the Walter and Eliza Hall Institute of Medical Research, and it's all various videos on different science issues. Restriction enzyme, EcoR1, molecular visualizations. So it's pretty cool. It's a nice. I had never seen this before. Thank you, Stephen. And that will do it. Oh, by the way, Steve says it's 2 degrees centigrade or Celsius here in the St. Louis, Missouri area. I think I just got this week, so that's about right for the Midwest. TWiV is done once a week, as you know, and you can find us on the weekend, usually Sundays, at twiv.tv on iTunes. And if you like us, do subscribe and leave a comment over on iTunes. Helps to keep us visible so more people can find us. And we'd love to get your questions and comments. Send them to TWiV at twiv.tv. Kathy Spindler is at the University of Michigan. Thanks so much, Kathy.
Thanks. It's always fun, and I learn a lot.
Us too. Rich Condit is at the University of Florida, Gainesville. Thank you, Rich.
Sure enough. Always a good time.
And Alan Dove is at alandove.com and on Twitter. Thank you, Alan.
Always a pleasure.
I'm Vincent Racaniello, and you can find me at virology.ws. So there you have it. A couple of professors and a science writer here on TWiV. Good mix. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.